Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Respiration in Plants. So in the last lecture we saw about the introduction. We saw the introduction of the respiration in plants. We saw what is difference between respiration and breathing, and then we saw that what are the different types of respiration, and then we started with aerobic respiration, in which we studied about glycolysis. And we also saw that the glycolysis is a universal site pathway which is present in all type of cells, irrespective of uh, whether it is aerobic or anaerobic, right? Then in the glycolysis, we saw that there are two phases, and the first phase was known as preparatory phase, and we covered this in the last lecture. We saw that in preparatory phase it comprises of the three steps, and these are the three steps, out of which two are irreversible and one was reversible. And we saw that this preparatory phase is a energy consuming phase, right? So it's an energy consuming phase in which the energy or the ATP is required to activate glucose, right? So here the glucose it gets converted into glucose 6 phosphate in the presence of ATP. So ATP was utilized and then it donated its phosphate group to this glucose to form glucose 6 phosphate. And then this glucose 6 phosphate undergo conversion to form fructose 6 phosphate. And then fructose 6 phosphate utilizes another ATP to get the phosphate group to form fructose 1,6 bisphosphate. And this contributed to the preparatory phase. Now, so what we studied about the preparatory phase is that in preparatory phase, we had utilized or the cycle pathway has utilized two ATPs. So it has utilization of two ATPs. Okay. Now the second is in preparatory phase, how many steps are involved? So as we have already written that there are three steps involved. Now the third thing about is that how many of these are reversible steps? Okay. So the number of reversible steps are how many? 3. And number of irreversible steps the number of reversible steps is 2. The total number of steps is 3, out of which the number of reversible steps, just a second. So the number of reversible step, if you see here, is 1. Okay, so the number of reversible step is 1, that is the isomerization step. And the number of irreversible steps are 2. Okay, that is first and third step. Okay. Now, apart from this, the another important thing is that here the enzyme that is involved in the third step, that is phosphofructokinase, right? So this phosphofructokinase is known as the phosphofructokinase. So it is known as pacemaker enzyme. Okay, it is known as the pacemaker enzyme. Okay, now what does pacemaker enzyme means? It means that it regulates the glycolysis. Pacemaker means it regulates the glycolysis. Okay, so it regulates the glycolysis, meaning it tells whether the glycolysis has to take place or not. So it regulates this glycolysis on what basis? So we know that the ultimate goal of glycolysis is the production of energy, or the ultimate goal of respiration overall is the production of energy, right? So what happens is that the glycolysis, when it happens, when does the cell know that it requires the glycolysis to take place? So it sees the amount of energy present in the cell. If the amount of energy that is present is adequate, then the glycolysis won't happen because the cell will not need ATP, right? But in the case there is deficient of ATP, in that case the glycolysis takes place, right? So what does this phosphofructokinase do? It does the exactly same thing. So it regulates the glycolysis based on the based on the ATP present in the cell, based on the ATP present in the cell. Okay, based on the ATP present in the cell. So based on this, it regulates the glycolysis. Okay, so this is about the preparatory phase. Now, before moving to the next phase, that is the payoff phase, let's see one important thing. So we know that this is an coenzyme that is known as NAD plus. Now, when this NAD plus is getting converted into NADH, now what this reaction will be called as? This will be called as reduction, right? So it will be called as reduction, right? Because this NAD plus is getting converted into NADH, addition of hydrogen. Now, so this is one thing. The second thing is, now you have this NAD plus along with another compound, say A. Now, what you are seeing is that it is A is converting to B, and this NAD is converting into NADH. So we are seeing that this NAD plus, it is undergoing reduction. So what we can say about this A? Okay, so what we can say about this A to B conversion? So if in a reaction, one substrate is undergoing reduction, it means the another substrate it is undergoing oxidation. Okay, so because we know that redox reaction it happens together, right? Here the electron that is getting accepted must be released from somewhere, right? So here the A is getting converted into B, and it is a oxidation reaction. It is a oxidation reaction, right? So NAD plus is getting converted into NADH, and NAD plus E is getting converted into B. So if one is getting reduced and the other is undergoing oxidation. Okay. Now the third thing. So this should be remember. Why we are seeing this, we will see shortly. Okay. Now the one way of representing the reduction is this. The another way is representing is NAD plus. If we have two electrons and two hydrogen ions, so this reaction will result in, now this NAD plus, if you see, it is already deficient of hydrogen ions, right? Sorry, electrons, because it has plus sign. So it will take an electron from here, and this will become your NAD, and now it will take a hydrogen ion and it will become H, and here will be another H plus. So this will be what will be formed when you will reduce NAD plus in the presence of electron and hydrogen ion. Okay. Now what is the use of this? We'll see shortly. Now we have studied about the preparatory phase. Let's move to the payoff phase. So what is a payoff phase? So payoff phase is a energy extraction phase. It is a energy extraction phase. Okay. It is an energy extraction phase. Now what happens in payoff phase? So we saw the last reaction, what we had formed. We have formed fructose 1,6-bisphosphate, right? So we have formed fructose 1,6-bisphosphate. Okay. Now this fructose 1,6-bisphosphate gets converted into subsequent compounds or products. Okay. And this phase, this payoff phase, it comprises of, if you see, 7 steps, because this is your step number 4. This is 5, 6, 7, 8, 9, and 10. So out of 10, the 3 steps involved in the preparatory phase, and the next 7 steps are involved in the payoff phase. Okay. So in the payoff phase, total steps are Total steps is 7. Okay. Now, what happens in the payoff phase? Payoff phase at the fourth step. So, step 4. In the step 4, what we are seeing that the fructose 1,6 bisphosphate is getting converted into a triose phosphate, which means that a 3 carbon containing phosphate, right? It will form a 3 carbon containing carbohydrate that have a phosphate group attached to it. This is meant by triose phosphate. And the name of this triose phosphate is what? Glyceraldehyde 3 phosphate. Okay. So one thing to remember that till now in the preparatory phase, everything was of 6 carbon compound, right? So in preparatory phase, all products are 6 carbon compound. 6 carbon compound. But in the case of payoff phase, whatever you will see, everything will be of 3 carbon compound. Okay. Meaning that this 6 carbon compound is getting converted into 3 carbon compound. So each of this compound will lead to the production of 2 molecules of 3 carbon compound, right? So what I'm trying to say, so initially you had 6 carbon molecule. Now it is getting converted into 3 carbon molecule. So if it is getting converted into 3 carbon molecule, how many molecules will be formed? 2, right? So 3 carbon containing compounds will be formed now on in the payoff phase. Okay. So in the step 4, what we have fructose 1,6 bisphosphate. So we know this is the structure of the first fructose. Here you have phosphate. Here you have phosphate, right? And okay. Now this fructose 1,6 bisphosphate, it is converted into glyceraldehyde 3 phosphate. So what is the structure of glyceraldehyde? So glyceraldehyde is a 3 carbon molecule where you have it is 3 carbon. Okay. Now it's an aldehyde, so it will have CHO group. It will have a OH group. Okay. And on the third carbon, it is a phosphate group attached. So instead of CH2OH, it generally have CHOH. Okay. But instead of OH, it will have now a phosphate group attached. Okay. A phosphate group attached to it. And because of this reason, it is known as glyceraldehyde 3 phosphate. Okay. Glyceraldehyde 3 phosphate. So what we are seeing here is that this phosphate that is here in the fructose, now it gets converted into, and it will be a 2 molecule of glyceraldehyde 3 phosphate. Okay. Now what happens? This glyceraldehyde 3 phosphate. Okay. And also the enzyme that catalyzes this reaction is known as aldolase. Okay. So the enzyme is known as aldolase. The enzyme is known as aldolase. Okay. And the third important thing is that this reaction is reversible. So this is a reversible reaction. Okay. Now, in the step 5, so whatever this glyceraldehyde 3 phosphate is formed, it is kind of unstable. Okay. So I'm writing it as a G3P, glyceraldehyde 3 phosphate. Okay. Now this glyceraldehyde 3 phosphate, it is unstable and it undergo isomerization. Okay. So it undergo isomerization to form what? It undergo isomerization to form another triose phosphate, that is dihydroxyacetone phosphate. Okay. So it forms dihydroxyacetone phosphate. Now we know that isomerization means that there is no change in the chemical formula. No change in the chemical composition, right? So the number of carbon, number of oxygen, number of hydrogen, everything will be same. It's just a rearrangement of those. Okay. So here it was aldehyde. Here it will form a ketone because acetone is a ketone, right? And this reaction is, of course, it will be catalyzed by isomerase enzyme because it's an isomerization reaction. And it is also a reversible reaction. Okay. It is also a reversible reaction. Now it's a reversible reaction. Then ultimately this dihydroxyacetone phosphate will gets converted into glyceraldehyde 3 phosphate. Okay. Now what happens? Now this glyceraldehyde 3-phosphate, in the step 6, this glyceraldehyde 3-phosphate, it it forms it forms another triose, but bisphosphate triose, that is 1,3-bisphosphoglyceric acid. So it forms, now this is the aldehyde, right? Glyceraldehyde 3-phosphate is aldehyde. Now it is forming the acid. So what will be the structure here? The structure here will be, because it's a 3-carbon, let's draw 1, 2, 3. And the third carbon, we have acid, it is saying. So we have COH. Here we had OH, this such as glyceraldehyde. So it's glyceric acid, but it will have OH. And then on the third carbon, we have H, and here PO, right? So like phosphate group. Okay, this phosphate. So this phosphate group, it's not like just P. It looks something like this. Okay, it looks something like this. Okay, so this is the phosphate that I'm representing by P. So P is equivalent to this thing. Okay, so this glyceraldehyde 3-phosphate, it gets converted into 1,3-bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid. Okay, so one thing to know. So here it is saying 1,3-bisphospho, right? So it has phosphate group at the third position as well as at the first position. Okay, so at its first position as well, it is first, second, third. So at first position as well, this phosphate group will be attached. Okay, so that's why it is known as 1,3-bisphosphoglyceric acid. Now what happens in this case? So this reaction is catalyzed by an enzyme known as dehydrogenase. Okay, and because it's phosphoglyceraldehyde, so the complete name of the enzyme is phosphoglyceraldehyde dehydrogenase. Okay. Now, what does the dehydrogenase do? So this dehydrogenase is used when you have another substrate, that is NAD. Okay, when you have NAD. Now, what we are seeing, that this NAD here, it is getting converted into NADH. Okay, it is getting converted into NADH. So just like I said in the last thing, that here we have two substrates, this glyceraldehyde 3-phosphate and then this NAD plus. This NAD plus, in the presence of this dehydrogenase, it's getting reduced, which means that this glyceraldehyde 3-phosphate, what is happening to it? It is getting oxidized, right? So this glyceraldehyde 3-phosphate undergo oxidation to form glyceric acid. Okay, so this is what happening in the 6th step. And the enzyme, whenever you have NAD plus, the enzyme that is involved here will be the dehydrogenase. Okay, and interestingly, this step is also reversible. This is also a reversible step. Okay, so this is the step number 6. Okay, where we have now formed 1,3-bisphosphoglyceric acid, and we have also formed one molecule of two molecules, because we know that everything that is forming in the payoff phase, it will be 2 molecules. Okay, so we have formed here from 2 NAD plus, we are forming 2 NADH. Okay, now next what we'll have step number 7. We'll have step number 7. So what happens in the step number 7? We have 1,3-bisphosphoglyceric acid. Okay. Now what we are going to do, or what the next reaction will be. So the next reaction will be, so the next reaction will be, we have 1,3-bisphosphoglyceric acid. Now the next reaction, it will form from 1,3-bisphosphoglyceric acid, it will form 3-phosphoglyceric acid. 3-phosphoglyceric acid. Okay. Now, how does this 3-phosphoglyceric acid looks like? Again, it's 3 carbon and we have glyceric acid. So it will look similar, just like this, except it will do not have phosphate group at the first position. Okay. So it will look just like previous one, but it will not have phosphate group at the. So this is your first, second, third. So it will have phosphate group only at the third carbon, not at the second carbon. Okay. So this is how it will look like. Okay. So this is 3-phosphoglyceric acid. And now what happens in this case? So in this case, the enzyme that is involved is known as phosphoglycerate kinase. It is known as phosphoglycerate kinase. Now the kinase is involved. And the second thing that happens, so if you see here the reaction, here we had two phosphate groups, right? One at the first position and another at the third position, right? But here what is happening? Here, one of the phosphate group is removed. So this reaction leads to the removal of one of the phosphate group. Okay. Now in the cell, the ADP that are present, they will take this phosphate. So ADP will come and it will take this phosphate and gets converted into ATP. Okay. So this is what happens here. So the phosphate that is released from 1,3-bis-phosphoglyceric acid, it is taken by ADP to form ATP. And this is known as substrate level phosphorylation. So this ATP formation is known as substrate level phosphorylation. Now, why it is known as substrate level phosphorylation? Because here what we are seeing that this substrate, the phosphate is getting released from the substrate, that is 1,3-bis-phosphoglyceric acid, and then it is taken by ADP. So the energy that is, or the ATP that is formed, it is known as substrate level phosphorylation. Okay. Now, what is the 8th step? So here we have 2 molecules of ATP formed. Okay. Now, the next step is your 3-phosphoglyceric acid that I am representing by 3-PGA. Your 3-phosphoglyceric acid, it undergo isomerization to form 2-phosphoglyceric acid. 2-phosphoglyceric acid. Okay. And so it undergo isomerization. Now the isomerization, if it is happening, the enzyme will be involved as isomerase. So we can understand what is happening here. Here, instead of the third carbon, the phosphate group will move to the second carbon, right? So here the phosphate group will now move. So it will look something like this. Let's say 2PJ will look something like this. Okay. So now we have 2-phosphoglyceric acid form here. Because it is isomerization, there will be no addition of anything, no subtraction of anything, just a rearrangement of the atoms. Now in the ninth step, what do we have? So in the 9th step, we have this 2-PGA, 2-phosphoglyceric acid. It undergoes certain reaction in which it releases the water. So it releases the water, and now what we have formed is known as phosphoenol pyruvate. So we have phosphoenol pyruvate. So what we are seeing that the water molecule is released. So here we have OH. So it will look, the structure of this phosphoenol pyruvate will look something like this. So we will have CS2. It is still the 3-carbon molecule. Okay. So this is a phosphate group because in 2-phosphoglyceric acid, let me draw the structure of 2-phosphoglyceric acid. What was happening? We had CS2OH, CHOP, and COH. Okay. Now out of this, your water molecule is released. Okay. So this OH and this H, it got released together. And what we have CS2, double bond COP and COH. So this is what is remaining. Okay. So this is phosphoenol pyruvate. And this reaction is catalyzed by the enzyme known as enolase. Okay. So this is a phosphoenol pyruvate where water molecule is removed. Now the last step is what? We have phosphoenol pyruvate. We have phosphoenol pyruvate. Now it ultimately leads to the formation of pyruvate, or you can say pyruvic acid. So pyruvate, or you can say pyruvic acid. Okay. This happens. Now the reaction is catalyzed by the enzyme known as pyruvate kinase. It is catalyzed by pyruvate kinase. And what happens in this case? So this, what we are seeing, that phosphoenol pyruvate has one phosphate group, right? But if you see the structure of pyruvate or pyruvic acid, it will look CS2 COH. Okay, so it will look like this. So if this your pyruvic acid looks like this, it means that the phosphate group is removed now. And if this phosphate group is removed, so this phosphate group will be utilized by the ADP. Okay, so this phosphate group will be utilized by the ADP to form ATP. And again, this is what? This is a substrate level phosphorylation. Substrate level phosphorylation. So what we are seeing here is that in the case of payoff phase, we had two substrate level phosphorylations, right? In which the ATP molecule is being formed. So this is the total payoff phase involving, and now with the help of the structure, we can see that where phosphate group is being added, where it is getting removed, why ATP is being formed, and why it is known as substrate level phosphorylation. Now, to summarize this payoff phase, what we can say that how many number of substrate level phosphorylation do we have? So number of substrate level phosphorylation that I am representing by SLP, that is substrate level phosphorylation. How many do we have? We have two substrate level phosphorylation. Okay, step in which NADH is formed. So step in which NADH is formed. So what is the step? So the step is, if you go back and look. So at what step NADH is formed? So this is a sixth step, right? So this is the sixth step. And which enzyme catalyzes NADH formation? So what was the name of the enzyme? The name of the enzyme was phosphoglyceraldehyde dehydrogenase. So it was catalyzed by phosphoglyceraldehyde dehydrogenase. Okay. Now, the steps in which ATP is formed. ATP is formed. So what are the steps in which ATP is formed? So if you see, one of the ATP is formed here at the 7th step, then the ATP is formed at the 10th step, right? So the steps in which the ATP is formed is 7th and 10th steps. Okay. Now we can see the what are both the enzymes of kinase, but depending on the substrate, the name of the kinase varies. Okay. Now in the glycolysis, the first enzyme. So the first enzyme to catalyze glycolysis. So we have if we have substrate as a glucose, then the first enzyme to catalyze glycolysis is what? It's hexokinase. Right? It is hexokinase. And the last enzyme to catalyze glycolysis is what? It's pyruvate kinase. It is pyruvate kinase. Okay. Now, what is the final product? What is the final product that is formed? So the final product is pyruvate, right? The final product that is formed of the glycolysis, it's pyruvate. Okay. What are the irreversible steps in this reaction? So the irreversible steps. So all of this step that I'm saying, so this isomerization, it is reversible. Okay. It is the reversible step. This enolase, it is also reversible step. Okay. This, in fact, this seventh step is also reversible step. Okay. So out of the complete glycolysis, the number of irreversible steps are only 3. Okay. So they are total of 3 irreversible steps. And that are what? That are first step, third step, and 10th steps. First, third, and 10th step. Okay. So these 3 are irreversible steps. So what are the first steps? And these steps are catalyzed by hexokinase. The third is catalyzed by phosphofructokinase. And the 10th step is catalyzed by pyruvate kinase. So what thing we are seeing that is common here is that all these irreversible steps are what? These are catalyzed by the kinases. Okay, that is pyruvate, hexokinase, and phosphofructokinases. Okay, and these irreversible steps are main regulator of the glycolysis. Okay, so this is about the glycolysis. Now, the last thing that we have to see in the glycolysis is the net energy gain. So, what is the total energy gain that we can see in this reaction? So, we can see that in from one glucose, we have one glucose molecule. From one glucose, what we know there are two phases. The first one is a preparatory phase. So, the first phase is preparatory phase, and the second is payoff phase. Right. Now, what happens in the preparatory phase? What we can see that in terms of energy, what we can see in the preparatory phase, we saw that in preparatory phase we have 2 ATP consumption, right? So in the preparatory phase, we are sort of 2 ATPs, right? So we have 2 ATP consumption. Okay. Now, in the payoff phase, what we can see? So we can see here that at two positions ATP are formed, right? At the 7th position and another 10th position. But from one glucose molecule, if we ask how many ATPs are formed, so here the reaction that we are seeing that from 1,3-bis phosphoglyceric acid, that is one molecule of this 1,3-bis phosphoglyceric acid, we have 1 ATP production is taking place. But we already know that in the preparatory phase, everything is forming in like 2 molecules of everything is getting formed, right? So here, at each step, 2 ATP from one glucose molecule here, 2 ATP will be formed. Here also 2 ATP will be formed. So because in the payoff phase, 2 molecule from one glucose, 2 molecules of products are forming in the payoff phase, right? 2 molecules of products are forming in the payoff phase, right? So in the payoff phase, what we will have? We have a 2-step ATP is forming. So we'll have 2 into 2 ATP. That is the net gain of ATP is 4 ATP molecules. Right? We'll have net gain of 4 ATP molecules. So, the gain of 4 ATP molecules in the case of payoff phase. So if we talk about the net ATP gain in complete glycolysis, so net ATP gain in complete glycolysis. So what do we have in complete glycolysis? Then what do we have? So we have 4 minus 2 because 2 is getting consumed, 4 is getting formed. So we'll have 2 ATP molecules. Okay, so in glycolysis, we have net gain of 2 ATP molecules. Okay, now if we talk about the ATP, then the second is in terms of NADH. So the NADH in the preparatory phase, we do not have any consumption or release formation of NADH. But in the payoff phase, what is happening? We have one NADH molecule. Sorry, one time at one step we have NADH formation taking place, right? And that is here at step 6. But again, because it is from only one glycerol dihydrate phosphate, so from 2 glycerol dihydrate phosphate, 2 molecules will formed. So here the net NADH will be 2. Okay, so the net ATP gain is 4, 2, and net NADH. Net NADH will also be 2 molecules. Now, why this NADH is important? Because we'll see this NADH ultimately leads to the formation of ATP. Okay, and depending on it, it leads to the formation of more than 1 ATP molecules. Okay, so a single NADH, it leads to the formation of more than 1 ATP molecule, that is around 2.5 ATP molecules. Okay, so this is the net gain, 2 ATP and 2 NADH. Okay, now let's talk about the anaerobic respiration. So here what we are seeing that the glycolysis is a universal cycle, right? That's true. So it will be present in the both aerobic respiration and in the case of anaerobic respiration. But if we talk specifically about anaerobic respiration, then what happens? So in the anaerobic respiration, in the last lecture, we are seeing the difference between the aerobic and anaerobic respiration. And we saw that the anaerobic respiration, there are various changes. The first thing that is important in anaerobic respiration is that we know that it takes place in the absence of oxygen. That is true, that we know. The second is it occurs in cytoplasm. Okay, it occurs in cytoplasm. But if you remember the schematic of aerobic respiration that I drew in the last lecture, so we saw that the aerobic respiration, a part of it takes place in the cytoplasm, while the another part takes place in the mitochondria. But the anaerobic respiration, it takes place entirely in the cytoplasm. Okay. Okay, so it takes place in the cytoplasm. Second, in this case, less number of ATPs are formed. So in this case, less number of ATPs. So the ATPs that are formed during the aerobic respiration, in anaerobic, the lesser number of ATP will be formed. Okay, so less number of ATPs are formed. And third, in this case, the anaerobic respiration is basically the partial or incomplete oxidation of glucose. Okay, so it is partial or incomplete oxidation of glucose. Incomplete oxidation of glucose. Now, what do I mean by partial or incomplete oxidation? So we know that the formula of glucose is what? It is C6H12O6. Now, but when it undergo oxidation, what is expected? If it undergo complete oxidation, so in the case of complete oxidation, since this glucose has 6 carbon, so each of this carbon should get converted into a carbon dioxide molecule. So this complete oxidation should lead to the formation of 6 molecule of carbon dioxide. Okay, it should lead to the formation of 6 molecule of carbon dioxide. But in the case of partial or incomplete oxidation, what happens? The less number of carbon dioxides are formed. Okay, so instead of 6 carbon, say if 4 carbon is getting formed or 3 carbon is getting formed, then it is known as incomplete oxidation. So here, less number of, or you can say less than 6 carbon dioxide is formed from one glucose molecule. From one glucose molecule. Okay. Now, also this partial and incomplete, these are two separate terms. Incomplete means what happens that in the final reaction that ultimately less number of carbon dioxide is getting formed. But partial means that in a particular pathway, although the carbon dioxide that is getting formed is less or no carbon dioxide is getting formed, but the substrate that will be formed at the end, the product that will form at the end, will ultimately go into another cycle and it will lead to the formation of carbon dioxide. Okay. For example, in the case of glycolysis, if we see, so in the glycolysis, we do not see the formation of any carbon dioxide. So it is the example of partial oxidation. Okay. Why? Because here this pyruvic acid is getting formed. There is no carbon dioxide, but this pyruvic acid will ultimately go to Krebs cycle in the case of aerobic respiration, and then it will lead to the formation of 6 carbon dioxides. Okay. So this is partial. This glycolysis is a partial because here no carbon dioxide is getting formed, but ultimately this pyruvic acid will lead to the formation of carbon dioxide. While the term incomplete means that the ultimately at the end, a less number of carbon dioxide will be formed. And these anaerobic respirations are the example of incomplete oxidation. Okay. So the partial oxidation, you can write glycolysis. And incomplete, you will have anaerobic respirations. Anaerobic respirations. Okay. Now, these anaerobic respirations are two types. The first one is known as lactic acid respiration. So as the name suggests, since it is a lactic acid respiration, so the final product that will be formed is what? Lactic acid. Okay, final product is lactic acid. Now, where does this lactic acid formation take place? So this lactic acid formation occurs in bacteria like lactic acid bacteria or lactobacillus. Okay. So lactic acid producing bacteria, that is lactobacillus. Lactobacillus. And apart from this, it also happens in muscles. It also takes place in muscles. Okay. Now, why it happens in lactic acid bacteria? Because we know in bacteria, we do not have cytoplasm. We do not have mitochondria. And why it happens in muscles? So in muscles, it does not takes place always. But if you remember, in the case of extensive exercises we feel cramp in our muscles, right? So this happens due to the accumulation of lactic acid. So what happens in that case? So when we exercise, we need our body need to produce energy, right? And for the production of energy, what do we need? We need oxygen, right? But in the case of exercise, the amount of oxygen that is required, we do not somehow get it that amount. Okay, and this leads to the movement of this respiration to anaerobic pathway. Okay, so in the muscles, the lactic acid formation takes place when there is less oxygen. Okay, specifically during the exercise. Okay. Okay. Now, what are the steps involved in the lactic acid respiration? So, so the steps involved in the lactic acid respiration is, the first step is, there are two steps involved. Two steps involved. The first step is glycolysis. The first step is glycolysis, which means that glucose will get converted into two molecules of pyruvate. Right? Glucose will get converted into two molecules of pyruvate. This happens. Now, what happens in the second step? In the second step, this pyruvate, it gets converted into it in the presence of 2NADH plus 2H plus. It gets converted into enzyme known as lactic acid. It gets converted into product known as lactic acid plus NAD plus. Okay. So here the lactic acid is formed. So from this is pyruvate. So from one glucose, we have two molecules of pyruvate, then two molecules of lactic acid and two molecules of NADS D plus will be formed. Okay. And this lactic acid also 3 carbon bound. So, and this takes place in the presence of dehydrogenase. Okay. Because here NADH is involved, right? So it takes place in the presence of enzyme known as dehydrogenase. Okay. So if you write the stoichiometry, what we have from one glucose, what we are having? We are having two lactic acid, two molecules of lactic acid, plus two NAD plus. And the net energy, if you talk about, so net energy. So if you see in the second step, step B, do we see any formation of ATP or NADH? The answer is no. So what will be the net energy gain? So the net energy will come from this glycolysis cycle. Okay. And if you remember, in the glycolysis cycle, how many molecules of ATP are formed? So the number of ATP that are formed in glycolysis is 2, right? So the net energy is 2 ATP. Okay. Will there be any gain of NAD, NADH, or will there be any loss of NADH? The answer is no, because we see that in the glycolysis, we saw the net gain of 2 NADH, right? So what we saw the net gain of 2 NADH, right? But if you see in the second step, that is when pyruvate gets converted into lactic acid. Okay. So we were studying about the anaerobic respiration, and we saw that in the case of anaerobic respiration, what? Just a second, there's some technical glitch. Okay. So in the case of anaerobic respiration, we are studying about the lactic acid respiration. So in the lactic acid respiration, we saw that the final product that is formed is the lactic acid. And this lactic acid respiration, it occurs in the lactic acid bacteria such as lactobacillus and in muscles. Because in muscles during exercise, we have less oxygen. Okay. And because of this, the respiration pathway switches to the anaerobic respiration. Now, in lactic acid respiration, the two steps are involved. The first one is the glycolysis, in which the glucose gets converted into pyruvate, two molecules of pyruvate. And the second step is where this pyruvate, in the presence of NADH, it undergo conversion to form lactic acid and NAD+. Okay. So the net gain, if we talk about, so here what we saw that the glucose it getting converted into lactic acid plus 2 NAD plus, and the net energy that is formed is 2 ATP. And from where this ATP is coming? This ATP is coming from the glycolysis. Okay. Because if you see during the lactic acid formation or in the second step, we do not have any ATP formation taking place. So the net ATP that is coming, it is coming from your it is coming from the glycolysis pathway where the two ATPs are formed. And the second thing is, if we talk about the gain of NAD plus or NADH, sorry. So is there any gain or loss of NADH? The answer is no. Because in the case of glycolysis, the NADH was forming, right? The NADH was being formed. But in the second step, what happened? That this NADH was getting converted into, in the presence of dehydrogenase, it gets converted into NAD+. So the NADH that was formed during the glycolysis, it was utilized in the second step where the lactic acid formation was taking place. And hence, there is no gain or loss of NADH. Okay. So this is what is happening in the anaerobic respiration, especially the lactic acid respiration. Now, the second type of anaerobic respiration is known as alcoholic respiration. Okay. So what happens in the alcoholic respiration? The final product is ethanol. The final product here will be ethanol. Okay. Now, in this case, the three steps are involved. So in lactic acid respiration, 2 steps are there. Here, 3 steps are involved. And what are these 3 steps? We'll see. But here, this alcoholic respiration where it takes place, so it takes place in yeast. Okay. It occurs in yeast, such as Saccharomyces cerevisiae. Right. That is Saccharomyces cerevisiae. Okay. Then we have plant cells. So in plant cells as well, the alcoholic respiration takes place during waterlogging. Okay, during waterlogging, the alcoholic respiration takes place in the plants. Now, like I said, it involves 3 steps. So what are the steps? So the first step that is present here is known as glycolysis. The first step is glycolysis. So what happens in the glycolysis? In the glycolysis, we know glucose, 1 molecule of glucose, it gets converted into 2 molecules of pyruvate. 2 molecules of pyruvate, right? And here, what do we have? We have 2 ATP, net gain of 2 ATP and 2 NADH, right? Now, the second step that is involved here is known as decarboxylation. The second step is decarboxylation. Now, what happens in decarboxylation? So in the case of decarboxylation, the 2 pyruvic acids, the 2 pyruvate that we are seeing, the 2 molecule of pyruvate, the pyruvate, it undergo decarboxylation, meaning removing of the carboxyl group, right? So what happens? Now it is pyruvate is an acid, right? It's a pyruvic acid. Now this pyruvic acid, it undergo decarboxylation, which means, and then it forms acetaldehyde. So what is acetaldehyde? CH3CHO. This is the acetaldehyde. So it forms acetaldehyde and release carbon dioxide. Okay, it releases carbon dioxide. Now it's 2 molecule of pyruvate, right, from 1 glucose. So you will have 2 here, 2 carbon dioxide here form. Okay, so it's called acetaldehyde. The product is called acetaldehyde, and this takes place in the presence of enzyme that is known as pyruvate decarboxylase. Pyruvate decarboxylase. okay, and a cofactor, that is magnesium ion. So what you see that this pyruvate that was 3 carbon, right? It is getting converted into acetaldehyde, that is 2 carbon molecule, and the release of 1 carbon dioxide. Now, in the third step, this acetaldehyde, this third step name, in this, the acetaldehyde that is formed, CH3CHO, that is formed, it in the presence of, because this is 2 molecule of CH3CHO, it in the presence of 2 NADH plus 2H plus, it gets converted into ethanol, that is C2H5OH, gets converted into ethanol plus water molecule. Okay, so the product that is formed here is ethanol, and the reaction that catalyzes this, the enzyme that catalyzes this is known as alcohol dehydrogenase. It is known as alcohol dehydrogenase. Now you can see that dehydrogenase is involved where the NADH is being used. Okay. So this is the complete reaction in the case of alcoholic respiration that involves 3 steps, where the first glucose is getting converted into pyruvate. The pyruvate then ultimately gets converted into acetaldehyde, and then this acetaldehyde gets converted into ethanol. Okay. Now, what is the final product is ethanol that we can see. Now, the net gain of energy. Talk about the net gain of energy. What do we have? So, throughout the reaction, what we are seeing that the 2 ATP was formed during the glycolysis and 2 NADH was formed, right? Here, no ATP is getting consumed in the subsequent steps, which means the net ATP will be 2. Can we say the similar thing about the NADH? The answer is no, because as in the case of lactic acid respiration as well, this NADH that is formed in the glycolysis step, it is being utilized in the third step where ethanol formation is taking place. So here there will be no gain or loss of NADH. Okay. And how many molecules of CO2 is formed? 2 molecules. So, and 2 molecules of carbon dioxide is formed. And because of this reason, this reaction is known as incomplete oxidation. Or it is incomplete oxidation, because instead of 6 molecule of glucose, instead of 6 molecule of carbon dioxide from 1 glucose molecule, only 2 molecules of carbon dioxide is being formed. Okay. So with this, we have completed the glycolysis and anaerobic respirations, in which we have saw 2 types of respiration, that is lactic acid respiration and alcoholic respiration. And in alcoholic respiration, the compound that is mainly formed is ethanol. Okay. So that's all for today. In next lecture, we will see more about the aerobic respiration, that is Krebs cycle, linked reaction and Krebs cycle. So that's all for today. Thank you.